to welcome our last speaker, which is Tilini De Silva. Tilini is pursuing her PhD in the Department of Veterinary Biosciences at the University of Melbourne, where her research focuses on developing microRNA-based high-throughput diagnostics for Q fever and Rickettsia infections in humans. Tilini's work aims to advance diagnostic capabilities for these important vector-borne diseases through innovative molecular approaches. Prior to that, she did her Bachelor of Science with honours in zoology at the University of Rickettsia, Sri Lanka. Please welcome Tilini. Thank you.
Hi everyone, thank you Annalise for the introduction. So very good evening to everyone. So first of all, I'd like to add an acknowledgement of the country. So I didn't think this would turn out like one of my supervisory meetings because one of my supervisors are here, I think. So today I'll be talking about my PhD project on microRNAs as potential diagnostic biomarkers for Q fever and Rickettsia. So before we begin, just to give a brief introduction, so microRNAs or miRNAs, they are small non-coding RNA molecules around 90 to 25 nucleotides in length. So they play crucial roles in post-transcriptional gene regulation and are involved in various other cellular processes. So in recent years, they have emerged as promising biomarkers for vector-borne diseases and several other diseases. But the research in this field is still largely exploratory, with very limited data available as of now. So the 2024 Nobel Prize in Physiology was awarded for the discovery of microRNA and its role in post-transcriptional gene regulation, and I was thinking at least I'm in the right field. So moving on, so just to give a brief introduction about my PhD project. So my PhD project involves identifying serum-based microRNA biomarkers for diagnosing Q fever and Rickettsia infections in humans. So very briefly, my experimental workflow includes collecting serum samples from Q fever and Rickettsia infected patients, and then method and protocol optimization. And then I have conducted an experiment to test the stability of circulating microRNAs in serum. And next, I plan to move on to the comprehensive profiling of microRNAs in my Q fever and Rickettsia case samples. And the next steps include biomarker selection using AI and machine learning, finally followed by the validation of these candidate biomarkers. So after identifying the knowledge gaps of the available literature in the field, I conducted a systematic review of differentially expressed microRNAs in humans and arthropod vectors with respect to different VBDs. So I basically started off by searching for peer-reviewed for databases for peer-reviewed articles, and following PRISMA guidelines and rigorous screening, I might add, and eligibility criteria, 79 studies were included in the review finally. And this paper is currently under review at PLOS NTDs. So this is a diagram of the breakdown of my study types. So most of the research was conducted on dengue, chikungunya virus infection, malaria, and JE, as you can see from the diagram. And also a considerable number of studies were conducted on human hosts and also on tick vectors. Moving on, so if you look at the geographic distribution of the included studies according to the host type, as you can see, most of the studies included in the review were conducted on ticks, mainly from China. And it is very important to note that there were no studies whatsoever conducted here in Australia regarding to vector-borne diseases. And if you look at the geographic distribution of the included studies according to the disease type or the VBD type, most of the studies were originated from Asia. And currently there are no in vivo studies on Rickettsia or Q fever conducted worldwide. So as I mentioned before, since most of the studies have focused on tick vectors and their microRNA profiling rather than any other vectors, so I came up with a diagram which illustrates the differentially expressed microRNAs involved in the tick life cycle from egg to adult. So the microRNAs that are marked in red are significantly upregulated, and the ones that are marked in blue are significantly downregulated. So as you can see from here, most of the studies have actually focused on microRNA profiling of nymphal and adult ticks as opposed to other life stages, because these stages are the most important and which are most responsible for disease transmission to humans. And this is another figure that illustrates the distinct microRNA expression profiling of tick organs. So like before, most of the studies have focused on the microRNA profiling of tick saliva and salivary glands, because they are the ones that play a crucial role in spread of pathogenic microbes to vertebrate hosts. And now I'll briefly share some examples of microRNA responses that I found interesting, which were associated with different VBDs. So there were 3 studies conducted on Chagas disease, and the cases with chronic Chagas disease cardiomyopathy showed an overlap of this particular microRNA MIR208A, which I found really interesting. And in the studies on African triminosomosis and babesiosis, these microRNAs were significantly upregulated and significantly downregulated. And moving on, in the case of schistosomosis, SMA MIR bantam, so this is a parasite-derived microRNA, and it was said to be significantly upregulated in both serum and extracellular vesicles of patients. And some of these studies have actually reported that microRNA biomarkers could sometimes offer advantages over serology for evaluating clearance of infection and also like detecting secondary exposure to schistosomosis. And when we look at Crimean Congo hemorrhagic fever, samples from fatal cases of that disease has shown a unique microRNA signature, as I have shown here, with an upregulation of these particular microRNAs and downregulation of several LET7 family microRNAs. And in terms of Rickettsia, this is an in vitro study that was conducted, and as I mentioned before, there are no in vivo studies at the moment, which was conducted in humans. So that concludes the systematic review part, and now moving on to some of the experimental procedures of my project. So I basically sourced my Q fever case samples from the Australian Rickettsia reference laboratory. So before starting the actual biomarker study, I actually had to address a very practical challenge. So since the samples that we get to ARRL are transported through various pathology networks, they can spend around like 1 to 5 days in transit. So some of these samples are shipped to us at 4 degrees with ice packs and all that, but the other samples, they basically arrive at room temperature, which is like 22 degrees. So to assess whether these transit delays do affect the stability of circulating microRNA, I conducted a pilot time series experiment using serum samples which were incubated at 4 degrees and also at 22 degrees. And I incubated them at different time points, and I used 3 biological replicates. And I must mention that previous studies have actually been limited to small PCR-based analysis, whereas this experiment uses a large-scale NGS approach to assess whether short processing delays affect circulating microRNA signatures. So this is the workflow of my stability experiments. So starting from RNA extraction to cDNA library preparation, and then next generation sequencing, and then NGS results analysis include read trimming, quality control checking, microRNA identification, and finally normalization. So now if you move on to the results of the stability experiment, so first we'll look at some graphs on data spread and data distribution. So these 2 graphs illustrate the distribution of log normalized microRNA read counts across different times. So as you can see, the consistent distribution across samples do indicate that the observed differences later, they are not due to any technical bias or anything, but they actually do reflect through biological variations such as temperature or time effects. So this actually confirms that our normalization process has been effective and reliable. So I have also performed statistics on this, and there are no significant differences here. So since manually looking at all the microRNAs was time consuming and very hard, so we developed an R-based mathematical model to characterize degradation trends in the very initial stages of my data analysis. So this is just the workflow of the the code or the script that I used. So it will first loop through microRNAs, and then it will log transform the read counts, and then a nonlinear regression model will run for each microRNA, and it will store the outputs and then later apply filtering rules. So I just included this to show what the script does. So it will basically load the libraries and the functions we need, and then it will import and prepare the data in the way we actually want. And then it will filter low expression microRNAs by retaining only the microRNAs with counts per million CPM, more than 1 in at least 2 subjects here. And finally, it combines all the results and then exports. So yeah, this is a part of the output that I got from the model. So it gives me like a basic idea as to how it looks like. So it gives me the coefficient of degradation for each microRNA for each temperature and each subject. And so this actually did provide me with an initial insight into degradation trends of each microRNA under different conditions. And after being able to look at that, I developed this analysis pipeline to categorize microRNAs into different categories depending on their behavior. So I started off with unfiltered 487 microRNAs, and then I removed them using the CPM filter, and then retained 198, and then I removed 9 of them due to missing counts, and finally I came up with 189 microRNAs for my downstream analysis. So in these 189 unique microRNAs, they were divided into decaying and stable based on a two-fold mean log2 change. So if the drop was more than like 1, it was categorized as decaying, and if the counts changed less than twofold, it was categorized as stable. So, and I must also mention that a small subset of microRNAs that did not clearly fit into both of these categories were designated as ambiguous and were set aside. So as you can see here, majority of the microRNAs were stable. That means 84% and 84%, 159 of them were stable up to 120 hours. And when moving on to room temperature, that is 22 degrees, still most of them were stable. 142, that means 75% of them were stable, while only 32 were decaying. And so this is an example plot of a stable microRNA. So y-axis is the normalized counts, and the time x-axis is the time. And since there were 3 subjects, the colors are for the 3 subjects. So there will be 2 plots for the same microRNA at 4 degrees and also at 22 degrees Celsius. So as you can see, they look pretty stable from 0 to 20-hour time period. So here I have included some more examples of such stable microRNAs. So they look pretty stable up to 120 hours. And these are some of the decaying microRNAs. So as you can see, when I went through them, I actually observed something. So I actually kind of saw that most of the decay happens at the first 24 hours, and then they look quite stable. And I was able to see this trend in a lot of microRNAs that were actually decaying. So after seeing that, to see if this observation holds true, I generated 2 heat maps just to see if that was true. So red indicates major decay and white indicates less decay. So as you can see at 4 degrees, most of the degradation also happens at 24 hours, and also at room temperature the decay is a bit more, but it's still happening at 24 hours. And this rapid decrease at 24 hours, I was actually thinking this could likely offer as free microRNAs are quickly degraded by exonucleases and endonucleases, while the microRNAs that are bound to proteins such as arganoate or the ones that are enclosed in vesicles remain protected and might remain stable. So that was my initial guess. So yeah. So these are 2 lists of the stable microRNAs and the decaying ones that I found. So yeah, in conclusion, the stability of microRNAs over 120 hours shows that reliable biomarker analysis is still possible even with short delays in sample processing or transport, representing a key advance for translational research. And finally, microRNAs do show strong potential as minimally invasive biomarkers for early diagnosis and disease tracking in BBDs. And finally, current research is still preliminary with limited validation across different aspects, which underscores the need for standardized studies in human and other vector contexts. Finally, I'd like to extend my sincere gratitude to my amazing supervisory panel, including Simon, John, Cameron, Ryan, and Nathan, and special thanks to the RRL team, including Stephen and Taka, for their invaluable support and assistance. Thank you so much.
Great talk. Thanks, Tilini. Do we have any questions? Oh, you can make me work for it.
Hello, yeah. Amazing talk. I was curious to know, because you found certain microRNAs were stable and few were degrading, even in the first 24 hours. Did you find any signatures, any common signatures of those microRNAs, which were degrading faster versus the one which was stable? Yeah, more from a sequence.
Yeah. So I think I got the question correct. So you're asking if there were different types of microRNAs which were degrading but more than different types. Well, it actually depends. So I'll go back. So that is the list of the decaying microRNAs, and these are the ones that are stable. I'm not very sure. So it might there might be something to do with the nucleotide length or the way that it's behaving or their physiology, but I don't have a very exact true answer that I could give you. Maybe.
Great talk, Tilini. Just a quick question. So, given these are very short sequences, how do you decide whether, how do you separate each sort of species of microRNA from each other? So, what's the criteria for single base pair difference, or how do you decide that?
I think for it to be called a microRNA, it should be short, rather than just RNA. It should be around, I think the general rule of thumb would be 19 to 25 nucleotides in length. That's what you're asking. And what was the other part? Oh yeah, sure. Yeah. So they are pretty short. Yeah.
Thanks, Tilini. Great talk, and my question might be a bit embarrassing considering that I'm on your supervisory panel. You're predicting, can you confirm, right, you're predicting or modelling the stability of these mirrors, and you've also done an experiment in the lab where you can go and validate those findings. That's correct?
Yeah. So the thing with the model was, it was just to give me an idea as to how they would behave. So when I first got the data, it was very hard for me to kind of dive into how to actually analyze the data. So the model actually helped me to plot the data, but I actually did the analysis by myself. So this part I did this by myself. So the model part was just to give me an idea. So it gave me the coefficient of degradation for each microRNA, so which gave me an idea as to how much one decays and which ones are not decaying and which one are decaying, and if there is a difference between different, because there were 3 subjects, if there's a difference between that. So yeah.
That's really interesting. Have you looked to the literature to see if there's any precedent or comparison of your findings compared to other people's findings? Because your findings would suggest that these molecules are remarkably stable for an RNA species.
Yeah, so to answer that question, so there was one honor student that was working in our lab. He did a PCR-based study. So he also basically did the same thing, but he he actually did a PCR-based study with the same experimental procedures. And here in my list of microRNAs, so I think Taka could confirm confirm this. So he also found that MIR 21-5P and 22-5P, they were also stable for up to 120 hours. So which kind of validated what I did. So, but in literature, I'm not sure if they've done I don't think they have done NGS, but in terms of PCR-based studies, that is the most recent example that I could give you.
I have a question. So in terms of what's next for your biomarker discovery for Q fever, is the plan to just omit those that you know are decaying, or can you use them as a sort of quality control marker in your qPCR?
Yeah, so I think that is like an added advantage for my biomarker study, so that we know that these set of microRNAs are degrading, right? But in like a sample collection point of view, we need to get as many samples as we could, because there will be a small delay when we get the sample. So I think we could use some of them to kind of make sure that the number of the reads that are in like a very reasonable amount of level. So yeah.
Any questions online? No? Any more in the room? Oh.
Oh yeah, thank you. Great talk, thank you. I was wondering, you showed some graphs where you had the different subjects and tracking the degradation.
Yeah.
Were there any microRNA, they all look pretty consistent in all these plots, were there any that you found that were really different in between the subjects, like certain subjects had a lot more decay or a lot more stability than the others?
Yeah, so as I think I mentioned this, I'm not sure. So I actually, in my analysis, I have set aside a small number of microRNAs as ambiguous because they didn't belong to any of the categories. Maybe they are, I'm not sure, so they could be due to some fault in the assay, or like when I did my extraction, something went wrong or something. So, but the ambiguous ones, they look pretty weird because they don't belong to any of the categories. So in some individuals, they are very high. Some individuals, they are very low. So that is why I kind of set those aside, and they were just around like 8% and 5%, if I'm correct.
Yeah. Wonderful, very interesting. Thank you.
This is very interesting. That's why I have a follow-up question. So you were talking about machine learning and selecting microRNAs. So are you planning to use machine learning to sort of predict the decaying or stability, or what's your target for machine learning?
Yeah. So sorry if I wasn't clear before. So the machine learning and AI, that part comes for the biomarker analysis, not for the stability study. So this was like a pilot experiment that I did to make sure that there is not much decay in the samples that I'm getting, because if I do the analysis, do the biomarker study straight away, what if something has happened to the samples if the counts are not enough? So that is just a pilot study, and the machine learning and the AI part, those are for my biomarker study in the future. So I think I'm getting help from Cameron and Ryan on that.
Perfect, thank you.
Thank you so much, Tilini, and thanks all three speakers. Let's give them a round of applause.